This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1209, recorded on April 11, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 9 degrees Celsius, 50 degrees Fahrenheit, and blue sky, clouds. Very nice after a couple gray, rainy days.
Ooh, look, 9 degrees C here in Chelsea as well. Cloudy. It's apparently raining. It's been raining for many days. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's currently 46 Fahrenheit, 8°C, overcast. It is fortunately, warm enough that the snow that was on the ground when I woke up this morning had melted pretty quickly. Gosh, it's April. We're sick of the snow already.
But you haven't had much snow lately, right?
No. It was a brief flurries that we got last night. I got up this morning. I looked out the window. I said, great. But then it was gone.
You didn't have to pull out the wovel.
No, I didn't need the wovel, didn't need the electric snowblower. So it's all good.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi. It is exactly the same weather that Alan has. So I also have 46 Fahrenheit, 8 Celsius, and gray and rainy and blah.
It's supposed to be a good weekend to stay inside.
I'm actually probably going to spend the weekend going to a museum. And I think, or at least tomorrow, going to a museum. And you know what? I think it's a good museum kind of weekend, too.
Yes, that's also an excellent idea.
Also, last weekend was a rainy weekend here as well. If you appreciate these programs, if you enjoy them, we'd love to have your support to produce them because we don't do advertising. And furthermore, for you, Microbe TV is a 501c3 nonprofit, so your donations are federal U.S. tax deductible. Go to microbe.tv/contribute. What's happening with ASV, Kathy?
Pretty much the same news as before. The deadline for ASV Cares and Kitty Corp is May 9th. I had been telling you the wrong date for a long time, which may have gotten some of you active sooner. But now that the concurrent workshop assignments are out, you can plan your day and figure out what hours you might need your kids to be at Kitty Corp. And you can also apply for ASV Cares if you're not going to use the on-site daycare. So those are May 9th, and then early bird registration for the meeting itself, which is, as we know, in Montreal, Canada, is May 12th. So you don't want to wait till after then because it costs more to register.
A little bit of news, just 2 items. First one is an article in MSN. Novavax in sell-off after RFK Jr.'s comments on COVID vaccine. This is just a bunch of bullet points, right? Because it's for financial people. So, Novavax, FDA declined to approve the Novavax vaccine to license it. It's been on EUA ever since the pandemic.
Do we know why they declined? Was it just they didn't have enough people to get around to looking at the data and then approving it? Or did they look at it and say no? I don't think it was the latter, but I don't know if it was the former.
I know, but I can't tell you.
Oh, okay.
No, not many other people know.
Okay.
So RFK says, we're looking at that vaccine. It's a single antigen vaccine. For respiratory illnesses, the single antigen vaccines have never worked. We're actually shifting our priorities to multiple antigen vaccines, and NIH is already working on a number of those. All right. Now this is just silliness at its height.
Yes.
First of all, RFK, I thought you said no vaccine works. Now you're saying multiple antigen vaccines work? Okay. But in fact, it's not true that single antigen vaccines don't work. The COVID vaccine works very well.
Right.
It saved millions of lives. Shingrix works well, single antigen. RSV, single antigen. Anyone else? Can
Influenza.
Yeah.
Influenza is just HA, right?
Well, yeah.
Or okay, you're right.
Some, depending on which one you're talking about.
Right. Yeah.
So I don't know where he gets this stuff from, and people listen to him. That's not true. There's not a reason to reject Novavax because it was EUA'd, it was used extensively, and all the papers Daniel did it induced antibodies as well as the mRNA vaccine, and it was equally protective against disease. So
it seemed like the only data that we didn't have yet was how durable it was, and that there was some thought that maybe it would be more durable.
yeah, that's what we were all speculating on, but I don't know what's happening here. So Novavax is like, hey, we got all the data you need. Why didn't you approve it? So it's very frustrating. Alan, you want to talk about dire wolves?
Yeah, this is just a quick follow-up to a side conversation we had a couple of weeks ago about the woolly mice. So the woolly mice were very, very cute. And I think we even talked at the time about how they weren't particularly a scientific breakthrough, but they were adorable.
Yeah, the only key thing about them was that they were adorable.
Yes. And Colossal Biosciences, the company that did that, just went viral again for supposedly resurrecting dire wolves, which they very carefully orchestrated the PR around this. There's no peer-reviewed paper, but probably all of our listeners heard this. It was on TV. They've got, cute little fluffy white puppies that they're showing off and saying these are dire wolves. They're not. They're gray wolves genetically engineered with a couple dozen gene changes that may or may not be related to characteristics of dire wolves. And I'm linking to a science article that goes into the details on this.
Did they use any frog DNA? Those in the know will understand that. Do you guys know what I'm talking about, Alan?
No, I don't.
I don't.
So, in the book, Jurassic Park, I forgot.
Oh, yes. Yes.
They, they didn't have all the. They got a little bit of dinosaur DNA from amber, right? From mosquitoes, and then the rest they used frog DNA.
Right.
Why would you use frog DNA? I don't know.
Of all things, yeah, no.
But the funny thing is, there's a sequence in that book, and if you blast it, it's just PBR 322.
Oh, and speaking of going through freezers, I just threw away some really pure preps of PBR 322.
PBR 322 is one of the original cloning plasmids.
Yeah.
Widely used tool in the lab.
Yeah, it was really useful. Had a couple of nice restriction sites and ampicillin resistance or tetracycline resistance, which you could use to select for colonies that received it.
I used it myself when I was in grad school.
I probably have used it.
Yeah.
And one more article in Reuters, also bullet pointed by Kennedy sets September deadline to identify cause of rising U.S. autism risks.
I'm very frustrated about this one.
Yeah.
I mean, this has been studied for decades. Okay. It's a complicated genetic problem. You can't put a deadline on it. And
you can't put a deadline on it, and the premise is wrong. the idea that
the deadline also. September?
Yeah.
I mean,
the premise is that there's some, recent tremendous rise in the incidence of autism, which is at best debatable, probably wrong. There's a rise in diagnosis of it because we've gotten better at diagnosing it. And if you look at the demographic data, you find that people who, are older who have it are more likely to have been diagnosed later in life because they weren't diagnosed as kids. But this is not some sudden change that's caused by some thing that's changed in the modern world, which is what RFK wants to try to say, that it's due to vaccines or, some other thing. And if we just went back to the way we were in primitive times, then everything would be better. Not the case.
No, everything would not be better.
No, nothing would be, hardly.
In fact, I did a lecture on measles in my emerging infectious disease class this week. My students are appalled. They can't believe that anyone thinks such a thing is a good idea.
All right. Today is Norovirus Day.
Yes.
I don't know. You guys remember the Dixon story about norovirus?
Oh, yes.
Mm-hmm.
So it used to be called Norwalk virus. And one TWiV, he said, oh, yeah, Norwalk, Connecticut. And people got all pissed off because it's Norwalk, Ohio, where it was first identified. The people in Connecticut didn't like it associated with them. Of course, Norwalk was gotten rid of. Now it's Noro.
I think it's just one actual isolate is still the Norwalk isolate, most likely.
It's still called Norwalk isolate, yeah.
Maybe.
That doesn't mean you can't get noroviruses in Norwalk, Connecticut. I'm sure you can.
But Dixon was always funny about that. He always said that he'll never forget that. Anyway, we start with a snippet. So today we're going to talk about potential vaccine and potential maybe therapeutic monoclonal antibodies.
And these are both related to the same underlying work. It's the same vaccine, right?
No, I don't think so.
Very close, if not
very,
the same.
very closely related.
Yeah.
I don't think so. I was actually, well, we'll get into it.
Okay. Yeah.
Okay.
It's a G1. It's a G1 vaccine.
Oh, right.
Well,
G1 in this. That's
actually, in that paper, they made G2 vaccine too, I thought. Or they referred to a G2 vaccine. Anyway, we're getting ahead of ourselves.
Yeah, so there are authors from the same company, Vaxart, on both papers, so it's closely related.
Okay. Kathy, you want to summarize this?
Sure. So I think there's only one author overlap between this first paper and the second paper. And it's the first author, Becca Flittner, who is a co-corresponding author here with James Cummings, and they're both from Vaxart. There's one author from University of Maryland, but everybody else is from this Vaxart company, which is in South San Francisco. And so this snippet is a report of a clinical trial producing a vaccine that might work against norovirus. And it's just the phase 1 trial, which is really just the safety trial. And this vector, or something like it, is what I had in my notes, was used in the patients that are used in the second paper that we're going to talk about. So the premise of this first paper is that immunizing adults for things in general is a challenge because their immune systems are waning. Immune system function declines with age.
Wait, you know what I really like about what you just said, Kathy?
Yes.
You said it was a problem in adults.
Oh, yes.
And does that mean I'm not an adult?
Older adults.
Older adults.
But then where do you draw the line for older?
Yeah. I'll take it.
Right. And norovirus itself is a challenge because there's many types to immunize against, and people have been trying. And there's, we'll talk more about this for the second paper. And in the United States, adults that are over age 65 are at the greatest risk for norovirus-associated mortality. So that's why they're trying out this vaccine that they hope will be a good platform for vaccines for other things in older adults, and in particular for norovirus. And so they develop a thermostable vaccine that's an oral tablet that's enterically coated and adenovirus-based. So it's going to be related to some of the adenovirus vaccines we've talked about before. It's based on an adenovirus type 5 backbone. It's a non-replicating vector, and they also include a double-stranded RNA adjuvant. And so this whole backbone and adjuvant and everything also expresses the norovirus VP1, which is the main capsid protein. And they give it to adults 55 to 80 years old. And it's going to, because it's enteric and goes through the gut, it's designed to release the vector into the intestinal ileum. And this previously was shown to be safe in people ages 18 to 49, and it generated cellular and humoral responses. And so now they're just trying it in an older age group. So randomized placebo-controlled trial, 63 adults completed the trial. There were only mild to moderate adverse effects, and the overall take-home message is that they produced this vaccine that safely generated strong, durable, norovirus-specific systemic, cellular, and mucosal immune responses. But again, this is only the phase 1 trial, so they have a ways to go with more individuals, etc.
Okay. So you did say most of the deaths occur in older individuals, right? Over 65?
Mhm.
Right.
Yeah, so the vaccine's got to work in them where, not all vaccines work well in older people.
Then,
They call it the vaccine efficacy gap.
Yeah. They say in the introduction 90% of norovirus associated deaths are in people over 65 in long-term care facilities particularly. So these are going to be people who probably have other issues too.
When we finish recording, remind me to tell you a story. I don't want to put it in the recording about norovirus. Okay. The vaccine efficacy gap. Mind the gap. That's what this is about. You guys know what mind the gap is?
Yes.
Oh, yes. Been there.
Sometimes people don't know. Like I said to my class this week, Bamlanivimab, the monoclonal, we used to call it Bam Bam. And I look at him and I say, does anybody know what Bam Bam was? And this one guy said, yeah, wasn't that a Flintstones character? I said, very good.
Yeah.
It's long before your time. So often I say things and I wonder if they get it because I'm increasingly gapped away from,
Yes.
from the students, right? Okay. So, we'll see this more in the next paper where we get some information. But there's so many genogroups and types of noroviruses. It's very hard to make a vaccine that covers all of them. And in this paper, they want to do a mucosal vaccine because, mucosal antibodies correlate with protection against norovirus disease, not infection,
Yes.
disease.
And it does seem particularly important because this is a mucosal entering pathogen and mucosal replicating pathogen, potentially a mucosally spread pathogen. So thinking about getting the mucosal immune response involved here instead of a systemic immune response is going to be quite important.
Yeah, but I'm going to tell you that IPV is a really good vaccine against poliovirus.
That's true.
but the difference is with IPV, we're trying to prevent invasion into the CNS.
Yes.
We're not trying to prevent gut replication. We don't care about that because once it gets in the blood, then it could get in the CNS. So that's why IPV works. You don't have to have a mucosal response.
Whereas with norovirus, the gut replication is the whole problem.
Yeah, the whole problem is two buckets. Gut,
yes.
throwing up and diarrhea. All right. So this vaccine is called VXAG 1.1. As Kathy said, it has a Ad5 vector, and then it's a G1.1 VP1 antigen with double-stranded RNA. So I guess it's all in the tablet. It's very interesting.
This is a really cool technology. They've got this tablet coated so it'll make it through the stomach into the intestine.
Yeah. And
then this thing uncoats. And then the adenovirus can presumably infect cells in the intestinal, epithelium. And then you get, since it's a non-replicating virus, it's going to go in, infect, produce the norovirus protein, give you the antigen, and they also have it producing this double-stranded RNA, which is a built-in adjuvant, which I just think the whole thing is very clever.
I don't know how it works, actually, because I view the intestine as a fast-flowing system, right?
Right.
And this tablet gets into the intestine, it dissolves. It's got to infect the intestinal cells, mucosal epithelial cells, right? Isn't the RNA going to get diffused away? I don't know how
the whole point of the intestine is that it's going to try and have contact with the whole contents of it.
Yeah.
Right. So, and it's not a pipe. It's
not a pipe.
It's flexing around
Synapasm peep.
Right. It's moving things through by peristalsis. So you've got all this movement.
No, I got it,
Vincent.
Don't worry.
Yeah.
There's a bagel store downstairs. There's a picture of a bagel, and it says Synapasm bagel. No, it's a picture of a bagel, Tower of Babel.
A bagel. Yes.
Yeah, I mean, it works. You're going to see they did a previous safety study, and it was fine. In this study, you're going to see it works, but I just don't know why.
Well, it gets in. I mean, it's infecting the cells the same way norovirus would infect the cells. Right? This is how norovirus gets in.
Yeah, I know, but this is a tablet with virus and adjuvant. And
yeah.
The adjuvant is in the vector.
The adjuvant's in the vector.
Yeah.
So the vector is going to make...
Yes. That's what I think is so clever about it. The vector,
Okay.
the vector produces the target antigen, the norovirus
that gets
antigen,
over that problem. Yeah.
and it also produces this double-stranded RNA right in the cell. The cell's going to produce that.
Is double-stranded RNA a TLR3?
It can be TLR3, yeah.
Okay, because that's what they have in this picture. I spent way too much time trying to find out more details about this vector, but I did eventually get one that shows an adenovirus with a transgene and the TLR3 agonist, which then I had somehow figured was the double-stranded RNA.
It must be an early promoter driving that, right?
They don't, so it looks pretty much like what they used to call gutless adenovirus.
Right.
That we all
It
hated.
has E1A, I think.
Yeah.
Okay.
Maybe. I spent too much time and still didn't get there, so I don't know. I'm not going to say what they have.
All right, so bring the adjuvant in the vector. That's the key. Very good. Okay, so this study, they have 65 people in the U.S., 55 to 80 years old, and they're randomized to get vaccine or placebo. We have, in the end, we have fewer people because people drop out. And the primary outcome is safety and tolerability. And then they're going to look at responses as well. So all the solicited events, right, the ones you ask about, were mild to moderate. Headache. Listen to this. This is funny. Headache, malaise, fatigue were the most common in both the vaccine and the placebo groups.
Yes.
Yeah. And about the same frequency.
Yeah.
Totally. So people get headaches. They get malaise and fatigue, whether or not you give. So these are not caused by the vaccine. And that's the thing. If it's the same rate in the placebo, then it's not vaccine related. Then 17 unsolicited treatment adverse events were reported within 28 days after the final dose, including one serious event of appendicitis, which of course was not related to the vaccine. But that's the thing; things happen to people, right?
Yep.
Mm-hmm.
So you have to. All these unsolicited adverse events were mild or moderate in severity. I didn't look at the supplemental figure. Does anyone know what they were?
I did not.
Didn't look either.
Okay, don't worry about it.
On that.
And again, the incidence of these unsolicited were the same in the vaccine and in the placebo group. So the summary is, when you get to this age group, people
Stuff
get
happens.
all kinds of things happening.
Well, I wouldn't even say when you get to this age group. I mean, everybody, you go out into life, and sometimes you get a headache, and sometimes you get,
Yeah, well,
you might get appendicitis. You might
after the first dose, did you have any of these things?
Right.
Yeah, I had a headache.
Yeah.
It gets written down, right? Which is the way it should be. This is a placebo-controlled trial, folks.
Yep.
So to say that no vaccine has ever been tested in a placebo-controlled trial is just false information, misinformation, and a lie, actually.
I just came up with a great idea.
What's that?
I think from my class in the fall, I think we're going to do a placebo-controlled trial of what things the class makes you do. I'm going to give my students and some other class like a thing.
Yeah,
Did you have a headache? Did you have this?
Yes.
And see what side effects my class led to.
Oh, also ask them if their teeth feel fuzzy.
Okay.
Ah.
Do your teeth feel fuzzy? Yeah.
Mm-hmm.
I mean, I think appendicitis is amazing, right? That this can happen in this. It's not a huge population, right? Okay, then they looked at serum antibody responses, IgA and IgG. And by the way, this is looking at days 1, 29, 57, and 210 post-vaccination. How many months is 210? A little over 6? Yeah, 6 would be 180, right? So like almost 7 months.
Well, it'll be 7 times 30, so.
Yeah.
7 months. Okay, so, the antibodies went up. And they do a comparison with, everybody has antibodies to noroviruses already, right? So they do fold rise over pre-vaccination baseline values for everyone. And the notable numbers, serum IgA remained elevated on day 57. And on day 210, both the high and medium dose cohorts had over twofold serum IgM over the baseline concentration, although these values were not significantly different from placebo. So, you expect these titers to go down by 210 days, if not sooner.
Yeah.
Right.
And it's also important to note that here they're looking at serum.
Yes.
And not at a mucosal site. They're looking at blood.
Yeah. And unfortunately, they don't look at memory cells at all in this paper, right? But, they say it lasts a long time because you're looking at months, but you're not going to have high titers 6 months and more afterwards. But you should have memory cells, right? But they didn't look at that. Okay. IgG also pretty much the same. Serum IgG, specifically anti-VP1 IgG, day 57. The IgG antibody was increased in the high, medium, and low dose groups compared with the placebo. It remained elevated on day 210. And importantly, and I didn't mention this for the IgA and the IgG, there's no difference in the age group. So they had two different groups, 55 to 65 and 66 to 80. And the responses were the same. So that says this is doing pretty well in the elderly.
Which is interesting because you kind of expect based on other vaccines, or I kind of expect based on other vaccines, you might see less response in the older people.
Yeah. Yeah, it's very surprising. Maybe if they had looked at T cells, they would see a difference.
Maybe.
And
we pointed out that this is dose dependent, right?
Yeah. Yeah, they'll have to pick what dose they want to use going forward based on this. Okay, so these are binding antibodies. What about virus binding? So the assay they use, they have virus-like particles, and they measure their binding, inhibition of their binding to a histoblood group antigen. All right. It's a surrogate neutralization assay because it's very hard to do neutralization assays with noroviruses. You can grow them in, gut organoid cultures, but not in your standard cell lines, so it's not so easy to do. And so this histoblood group antigen binding to virus is relevant to infection. So if you can block that, the antibodies may be important, right? And so again, they see increases in antibodies that block this interaction on day 29 and day 57. No differences in the two age groups either, 55 to 65 and 66 to 80. So they think that this antibody gives you good functional antibody responses, again, in serum. Right. They also look at the avidity of the antibody, the binding strength over time from day 1 to 29, and goes up, excuse me, it goes up as you might expect it to do. They want to know, in these experiments, they're not distinguishing between IgG and IgA. So they want to know which one plays a greater role in these blocking assays that they're doing. So they actually purify IgA and IgG separately from the sera of some of the participants. And then they do a neutralization, a surrogate neutralization titer, and normalize it per microgram of IgA. So for total antibody, and for IgA, the number is 2.8. So for IgG, it was 0.7. So looks like IgA is making a greater contribution to neutralization than IgG.
Which is reassuring.
Mm-hmm.
Is that fair to say from this, Brianne?
Yeah, that's what I think I would say.
Okay.
And it is reassuring, and it sort of goes with this idea of using perhaps a mucosal vaccine. it makes you think, okay, perhaps we are getting a mucosal response, as IgA is often thought of as a isotype that is often found in the mucosa, even though they're not measuring the mucosa here.
Right, serum again, yeah. All right, then they want to know if this vaccine can generate B-cell responses in old people. So they quantify antibody-secreting cells in the blood, and what they find is antibody-secreting B cells. They would be, and they quantify them in the different age groups, and there's no difference. They make the same numbers of antibody-secreting cells in both.
And they do also see another dose-dependent response.
Yeah. Okay. They say antigen-specific mucosal homing plasmablasts have been investigated as a proxy marker of mucosal immune protection. So the Brianne, what's a plasmablast?
So a plasmablast is an activated B cell. It is now sort of become more like a plasma cell. But the most important thing here is that this cell has proteins on its surface that help it travel to mucosal sites. So we know that there are certain proteins on the surface of lymphocytes that mean that when they're in the blood, they're getting ready to go to the mucosal sites. And since it's often can be challenging to actually get a sample from a mucosal site, and people often do what they do where they're getting serum samples, they will look in the blood to see how many of the lymphocytes have these proteins that mean that those lymphocytes are heading towards a mucosal site.
Okay. So they find that the IgA producing plasmablasts are in circulation, and they have the ability to migrate to the gut mucosa.
Yeah, so it's sort of like we've activated B cells, and the B cells that we've activated have the capacity to go to the mucosa. So perhaps we are getting a mucosal response here.
Next, they looked at T cell responses, and they find activated CD4 and CD8 T cells with the right marker, which went up in the circulation a week after oral vaccination. All right, so finally, is it in the mucosa? So they look in saliva. So they find that salivary VP1 specific IgA went up in all vaccine dosage groups, and on day 210, the salivary IgA remained elevated compared with placebo, but it's not huge. It's a little bit. No differences in salivary IgA between the age groups. So they say, this vaccine can give us long-lived salivary responses. Are there B cells in the oral mucosa, Brianne?
In the oral, I would say, yeah, I believe so. There definitely are B cells in places like the tonsil.
Yeah.
And some of the lymphoid tissue that is in the malt and nault.
Okay, so that could respond to
absolutely.
Infection, right? Okay. Another place they look besides saliva is the nasal lining fluid. So these are distal mucosal sites, right? Because they're immunizing the gut.
Yeah, I was thinking about that. And I mean, part of the problem is that getting samples, at least directly, of the small intestine would be tricky. Obviously, you can get some samples lower down a little bit more easily. But I would also imagine that if you really think about it, and you think about norovirus infection, the virus is probably coming in through the mouth, through ingestion. Yep. And so you want to have antibodies that might be able to neutralize pretty early. Sure. So this is I was thinking that would be a good reason to perhaps check this early, and it's just a site you can get samples from pretty easily. But this would be a good place for neutralization to happen in a perfect world.
Also, they also cite some earlier research, I think it was in the intro or the discussion, that apparently IgA, anti-norovirus IgA in the saliva correlates with immunity.
Yeah. And we also know that when you immunize one mucosal site, sometimes you get responses at other mucosal sites, because that's what you would want to do with this vaccine. You immunize the gut, and then you get antibodies in the saliva or the nasal lining fluid. And so this is basically nasal secretions, not saliva. That's different, right? So they measure nasal VP1 specific IgA. It goes up in all groups 1 month and remains elevated. To day 210, they're still higher, but only in the high dose group. So you can also find it in the nasal wash. You get antibodies in the nasal wash. They finally...
Right, so they looked at both saliva and nasal wash.
Yeah, both the saliva we already did. Now nasal wash is pretty much the same. Yeah. And finally, they have this idea that VP1 specific IgA positive circulating antibody secreting cells, which they see a week after vaccination, are probably associated with subsequent increases in IgA responses that they see. And so they look at peripheral ASC frequencies a week after and a month after post-immunization. And in fact, they go up. So they say these peripheral ASCs that we induce with our vaccine may be early plasmablasts with the capacity to move to mucosal tissues and become local antigen-specific secretory IgA-producing cells. So overall, this looks pretty good to me, my untrained eye. It's a orally delivered vaccine. It induces antibodies, not only in the serum, but in the upper respiratory tract, especially IgA antibodies that can interfere with norovirus binding. There's not a... Being old doesn't hurt you here. You do as well as younger people in terms of responses. And so they say that we gotta go on forward. But this is only monovalent G1.1 in this paper. They say we need to look at other... and they said, in fact, the larger phase 2 is ongoing, which uses G1.1 and G2.4, right? Well, the results in this paper are G1 specific. So we're going to hear more about this, I'm sure.
Yeah.
Now, this is a good question now. No, it's not a good question. It's just a question. So who's going to get this vaccine? Assuming it's licensed.
Depends on pricing, depends on, I think the priority would be people who are older people in long-term care facilities, because that's who's most likely to die.
They should all get it.
Yeah, I mean, I think that with some of the data that we had, especially with the COVID vaccine, it looked like a higher population percentage of people being vaccinated really helped some of those at-risk communities as well. And so I wonder if worrying not so much about age group, but populations, and maybe populations in congregate settings. So people in those long-term care facilities, maybe people on college campuses, maybe kids in daycares, places where you could imagine a lot of transmission happening. If we could block transmission in those sorts of settings, that might be protective of most of the high-risk people in the population.
And I think it makes an enormous difference that this is an oral vaccine. Which easy. I personally suspect is going to get around a lot of vaccine hesitancy.
Yeah, I agree.
It certainly drastically simplifies the logistics of distributing this thing, because anybody can hand out pills, and it's a, you can mail this to people. It's a stable tablet.
The diagram I did find about the vector itself shows the shipping is just in a pouch and says self-administration. And then I'm laughing because the last part of the picture is two stick figures. One is a a guy in purple, and then the other is a woman in green, and they're both dancing, and she's got a cane. So
that's good for all ages.
Yeah. Anyway.
That's good.
One more thing. What was I going to say here? Oh, it's gone. Well, these are probably going to be at least bivalent vaccines going forward. So that's good because what's-his-face won't object to them.
Right.
They'll have multiple antigens.
Yes, multiple
antigens.
Yeah.
I mean, don't all of them really have multiple antigens? Because
Yeah.
or at least if we're talking epitopes, probably more than one
wait a
epitope
minute.
in the antigen.
Are you expecting him to know what the hell all that is?
I know.
Oh my gosh. Epitope. You go up to him and say epitope, he'll put you in jail.
But if I just started talking about epitopes, then I would be like, see, it has multiple. It has multiple thingies.
Yeah, what did he call it? He had single what? Single. Jeez. Let me go back to the single antigen, he calls it. Is that what, did he use the word single antigen? The single antigen vaccines
Someone taught him the word
worked.
antigen?
That's even worse. Single antigen. I mean, I think he means like a single protein, like a
Right.
spike, right?
Right.
But that's many antigens in
Well,
that
it's
spike.
many epitopes, perhaps, in an antigen.
Yes.
Yeah, I
So,
suppose.
so it's all multiple.
This is very frustrating for science.
Yeah.
Oh boy. Okay. We have another norovirus paper for your digestion. Kathy, can you summarize that for us?
We all studied it, and now we're going to regurgitate the details.
The title is Broadly Neutralizing Antibodies Targeting Pandemic G2.4 Variants or 7G2 Genotypes of Human Norovirus. And there are 3 co-first authors, Jun Park and Lisa Lindesmith, both from University of Texas, Austin, and Adam Olia from this VaxArt. And then there are 2 co-senior authors, George Georgiou and Ralph Barrick at University of North Carolina at Chapel Hill. So, there's also contributions from NIAID, the Vaccine Research Center, that is, the Centers for Disease Control and Prevention. I can't read my writing. Oh, Utrecht University and the Frederick Cancer Center, and that's an NIH thing. And then, as I said, the VaxArt in South San Francisco. So, this paper, in very short terms, uses really highly sophisticated techniques to identify antibodies that will neutralize a broad range of norovirus variants. And so, again, to reiterate, this is a really bad infectious gastroenteritis. 700 million infections worldwide, causes tens of thousands of deaths. And again, the problem with getting a vaccine is the genetic diversity and then a lack of knowledge. And it's that knowledge gap that they're working on filling here. And the knowledge gap is really about what are the conserved epitopes that the antibodies will bind to and neutralize the virus. And they make a point that there are some suggestions that people do make some long-term protective immunity to noroviruses. For example, young children rarely get repeat infection with the same genotype or variant norovirus. And there's a lack of infections in adults in years when there are not new strains emerging. So that's some evidence that, yes, we could conceivably make some good vaccines if we had the right antigens to elicit antibodies. So how to do this? So they took advantage of the fact that there were immunized people that had been immunized with this adenovirus-based vaccine that expresses the norovirus protein VP1 and also has this RNA, double-stranded RNA adjuvant. And I did misspeak. The first paper, they did only use the genotype 1 and genogroup, whatever they call it, 1. And here it's genotype 2 that they're using. And so they take advantage, they immunize the population, and then they screen this population and find serum from 2 recipients that had robust responses to the vaccine. And one of them elicited antibodies that could neutralize several viral variants within a genotype. And the other individual developed antibodies that could neutralize across genotypes. And so, and these individuals were somewhere around 37 years of age, so not this older age group that the previous paper had. So they chose these two people and then characterized their the monoclonal antibodies from them. So they used a variety of techniques that we'll discuss. And the bottom line is that they identified some of these antibodies, and one in particular called VX22, if you want to remember one of them. VX22, it could block more than 7 genotypes of the major class, the G2 genotype. They used an approach called, with a set of words where I knew all the words but had never heard them together before, antibody evolution trajectory analysis, which is that they used phylogenetics to identify the antibody pathway to get particularly to this VX22 antibody. And then they used crystallography as another analysis to identify a highly conserved cross-neutralizing epitope that altogether this points to good targets for design for future antigens to produce vaccines. And they show that, at least their evidence points to the way that VX22 is working is by disrupting the virus particle, so causing it to disassemble such that it can't cause infection. And then another term that they use that I had not really heard before, and I want to hear the other people's take on what it means to be doing back boosting. How does that differ from just doing boosting? But I think they're trying to connote something by calling it back boosting. But anyway, so again,
it's not putting a pillow in your chair.
Right. The bottom line is that they've really used this technology to guide the design of immunogens that could make broadly protective norovirus vaccines.
There's that word, and there's another word that'll come up. We'll get to it. Okay, so just to remind everyone why we care about norovirus. Globally, 700 million infections a year, tens of thousands of deaths. Okay, so it's substantial medical burden. They're divided into genogroups. At least 4 infect humans, and genogroup 2 causes more than 90% of infections. Okay, genogroup 2, remember that. That's the big one.
And just as far as why we care about norovirus, they talk about this in the other paper too. Even without the deaths, the infections are not pleasant.
No.
Yeah.
We don't have enough buckets for everyone.
Exactly. And this is something that will, I think most people have probably experienced it, whether they knew it or not. I mean, the most common foodborne disease, or most common foodborne virus, certainly. So you eat out at a restaurant, and a couple of days later, you can't go more than 10 feet from the bathroom. And that's the experience of a norovirus infection. That'll go on for a few days, and you probably can't make it to work if you need to get out somewhere. It just completely hampers everything. So I think one of these papers cited a figure of direct medical costs in excess of $1 billion a year just in the U.S., and that's just direct medical costs. That doesn't account for
10.6 annually.
10.6 billion. And that's just direct medical costs. So that doesn't account for all the missed work and the, other stuff and the buying extra buckets.
So it's the biggest cause of gastroenteritis and of any bacterial, viral. It's the biggest one.
Yeah. So this virus is bad news. We'd like to be rid of it.
Okay. Genogroup 2, remember, over 90% of infections. And there's a genotype G2.4 that does 50 to 80% of the outbreaks. And this varies. So you get variants and they cause new outbreaks because they're antigenically different, right? But as Kathy said in her summary, you don't get repeat infections with the same variant. So that means there's some hope for preventing infection with a vaccine. All right. So I don't know if we mentioned this, but when you make VP1 of this virus, it assembles into a virus-like particle, right? VLP. And that's what's happening with these oral vaccines. They're delivering the gene into cells and they're assembling, presumably, into VLPs. And these are immunologically indistinguishable from native norovirus particles. Now, the problem with norovirus is not easy to do neutralization assays and identify the epitopes on the particle that react with antibodies, because you can't grow them in cells in culture for the most part, except for intestinal enteroids, which just doing a lot of neutralization assays is just not
a convenient system to work with.
And so they do use surrogate neutralization assays, like we said in the other paper, to look at virus binding to blood group antigens and other ligands as surrogates. So they say 5 norovirus vaccines are in clinical development, including a G2.4 one. So they can do 2 things. They can give you VLPs. I don't know how they would do it, maybe in a tablet also, or the adenovirus vectors that encode the VP1, which will make the GLPs. Oh, I see. In adults, the VLPs are given intramuscularly. And so they don't work so well. Okay. We need to do better.
And that could be a mucosal versus systemic delivery thing.
And they say we don't really know much about the antigenic structure. We only have a few neutralizing monoclonal antibodies, and we know their epitopes. And they give this example. It's very interesting. G2.4 Hong Kong 2019 variant emerged with antigenic changes in at least 6 known epitopes. That's a lot. 6 known epitopes. And no human monoclonals have been reported to neutralize this HK 2019 variant. So even though it hasn't spread extensively, they're worried that it will, because it's so different antigenically. And furthermore, no antibodies of any type that neutralize more than 2 G2 viruses have been reported. So they say we need to better understand the antibody response to guide vaccine design. So that's the rationale for this paper, essentially. And so what they do is they have 2 healthy young adults, and they immunize them with a single dose of an Ad5-based oral vaccine, similar to the one we talked about, except this has got VP1 of G2.4 SY 2012. So this is from a ongoing clinical trial.
Yeah, so different genogroup, G1 versus G2, and this one is G2, and it's that particularly frequent genogroup and particularly frequent strain.
Right. Now, but this is the same company, same platform. It's the tablet targeting the ileum, yada, yada, yada.
Yada, yada, yada.
Adenovirus vector.
So just to make it clear, I think that reading the materials and methods, they had access to this population that Vaxart had immunized, and they screened a bunch of them and picked these 2 based on their responses.
Right.
Yeah, that could be.
Yeah.
This was not 2 random individuals.
No.
Right. The results make it sound that way, though.
Yes. We just grabbed 2 people and they had these great responses.
We immunized them and then did this study.
Yeah, I mean, it says 2 donors were selected for detailed studies because of exceptionally high G2/4 neutralization breadth, participant A, or cross G2 neutralization breadth, participant B. So
A and B are dealt with separately because they have different findings with both.
Right.
And so in A, the vaccination boosted the ligand-binding blockade serum. Remember, this is the surrogate neutralization assay, to all tested G2/4 variants circulating from 1987 to 2019. What a pain that you have to circumscribe it so much. And then the participant B had exceptional cross-genotype blocking serum breadth across 7 G2 and 2 G1 genotypes before he was vaccinated. Oh my God, this person must have been a world traveler. Got all these antibodies, although maybe not. Maybe you got one good infection.
Yeah, maybe he had one good B cell that made some antibody that was very, very cross-reactive.
They're both females, and A was 28 and B was 37.
Then she is a world traveler.
Right.
You know, the interesting thing is the participant B, all these antibodies were modestly boosted by vaccination.
Mm-hmm.
It's very interesting. And they say there's probably immune imprinting going on, which we have evidence for, because they say in both donors the greatest fold increase in blockade titer was observed against the vaccine strain, whereas the highest titer was to the ancestral G24 variant, and they say that's probably immune imprinting. So the first norovirus you see, that's what you respond to with subsequent challenges, unless they are different enough, and how different we're not sure. Okay, so then they go on to, they say we're going to deconvolute the antigen-specific antibody repertoires. Well, basically, they want to know what antibodies are binding to what epitopes in these individuals. But they do it, they use bottom-up liquid chromatography, tandem mass spec, guided by a donor-specific mass spec search database comprising over 1 million heavy chain variable transcripts and single-cell VH light chain variable domain paired B-cell receptor reads, which they got from peripheral B cells on day 8. So these individuals, they make these libraries from them and get all of these reads out of them.
Yeah, so basically they found exactly what the DNA of those individuals looked like, and then they compared the protein fragments to make sure that they were getting a match of the actual DNA sequence in the individual donors.
Right. So this way they could determine the compositions of these antigen-specific IgG clonotypes and their abundance in serum. All right. So they purify the antibodies using immobilized VLPs, and then they sequence the immunoglobulin gene sequencing. They say this revealed an oligoclonal and polarized anti-G2.4 serological repertoire, with the top 5 most abundant clonotypes constituting more than 60% of the anti-G2.4 vaccination repertoire. And then they comparing the pre- and post-vaccination serum repertoires, so they compare this before and after vaccination. That vaccination back-boosted all antigen-specific clonotypes that were present at day 1. So, Brian, it's just boosting. It's just memory, right?
Yeah.
I don't know why you called it back-boosting.
I don't really know why they're calling it a back boost either.
I think they're just trying to emphasize that these were pre-existing, that this is a memory response, but it's a little convoluted.
Yeah.
Isn't boosting always?
Boosting is always
something
that was there already. Yes.
Yeah.
You don't need both words. That's true. Anyway, so vaccination increases the antibodies that are all present, the clonotypes present at day 1 already. And these vaccine-boosted antibodies comprise 65% of the post-vaccination serum responses. So really, I don't know how universal this is because it's 2 people, but I mean, I suppose you're going to at one point immunize people who have never seen norovirus. Is that possible? Nah.
Babies?
Are you going to immunize babies?
I don't think you're going to immunize babies with
this. So it's going to be older people.
Well,
Yeah.
is it really severe in young kids?
No. Well, I don't know. Babies, it depends where you are.
Right, because that might be a reason to immunize babies.
Could be. But
well, we know just from this paper and the fact that they picked these 2 out of a group that this is not the usual response.
Yes.
Yeah.
Yeah, I mean, you're going to have a de novo. So, Brianne, we did a paper recently where they called it a de novo response.
Yes.
And you said that we usually call it what?
I would call that a response from a naive cell.
Yeah.
Something.
Naive. Yeah. So, and I don't know. I mean, the point here is that they're using these as tools and they get some interesting antibodies. So it doesn't matter.
Right.
What they had before. But, I mean, if you want to figure out a way to induce these antibodies in a naive person, then that's a different story.
Yeah, I mean, there are a couple of ways that people think about those types of things, but before you can get that complicated, you need to know what is the target, like goal antibody, and what is the antigen that antibody binds, so that you can try to use that as your immunogen, and get this goal amazing antibody. Yeah. And it might be more complicated than that, but you at least have to know what the goal is and what the antigen is before you can do anything else complicated.
Yeah, if you don't know what you're aiming for, you're not going to hit it. And this paper is trying to figure out what to aim for, I think.
So they look at the antibodies of participant B to try and find antibodies that have broad G2 activity. And they have a way of doing that, which I'm not going to go into.
Because participant B was the one who had like neutralization of all sorts of different strains.
So they find that vaccination of B increases the abundance of G23 and G24 clonotypes by more than threefold on day 29 compared with day 1. And these account for over 75% of their serum response, those two clonotypes. And a big fraction of those were boosted by vaccination. I'm going to take out the back. Boosted by vaccination, which they say suggests recall responses from memory B cells. Yeah, that's what it is. Recall memory B cells elicited by earlier exposures. For participant A, okay, here's the other word, Kathy, that bothers me. 13 monoclonals, comprising 70% of the anti-G24 serum repertoire, were recombinantly expressed. Do you like recombinantly?
No,
I
understand what it means.
I know what they mean.
Yeah.
I mean, any kind of expression that they're doing here, they had to do by recombination.
Just recombinantly is a funny word.
Yeah.
All right, anyway. So then they characterize these antibodies. 10 of them blocked at least one G24 variant, and none of them blocked non-G24 strains. And they picked one of them, which was 44% of the repertoire, and they characterized it more in more detail. And they say that the blockade serum landscape of participant A was substantially driven by a single clonotype, VXI, which comprised over 40% of the total response.
VX1.
VX1, yes. Hard to distinguish.
They're so small.
Yes.
So then these G24 neutralizing antibodies, they put them in two classes. Class 1 neutralizes multiple G24 variants, except for HK 2019. And Class 2 neutralized diverse G24 variants, including HK 2019. So I guess the only difference is HK 2019.
Yeah.
More or less. Yeah. So these monoclonals recognize a site more conserved across G2.4 than Class 1 BNAbs. Class 2 recognize a site more conserved than Class 1 because of the HK 2019.
Yeah, so basically Class 1s recognize everything but HK 2019, so they recognize something that's on all of them, not HK 2019, and the other ones recognize something that's on all of them.
Then they have a collection of VLPs with amino acid changes in each of the antigenic site. They call them epitope mutant VLPs, which I totally disagree with. They're not epitope mutants.
You can't mutate an epitope.
No, you cannot. Don't argue with me.
You can mutate the gene that encodes that epitope.
Yes, exactly.
You could alter an epitope.
You could alter an epitope, yeah. And so they can identify, where these monoclonal antibodies are binding, the class 1s, for example. But I'm not going to tell you because it's not going to mean anything to you. Then they want to know the class 2 epitopes, but they had to make more VLPs. They call them G2 chimeric VLPs. At least they didn't call them epitope mutant VLPs. And they could identify the epitopes for class 2 as well. All right. That's all I'm going to tell you about that. Now, what about, can these antibodies block replication? That's a good question, right? We've just looked at this surrogate binding assay so far, ligand blocking. And so they select 4 antibodies, VX1 and 14 from class 1 and VX6 and 10 from class 2. Oh, I guess VX is Vaxart. Yeah, that's cool.
Ah, okay.
By the way, I like Vaxart as a company name.
Yeah,
I do like that name.
It's really good. Good job. And they use their organoids, their enteric organoids, to measure blockade, and they use PCR to measure genome copies, and all 4 monoclonals reduce the genomic copies of G24 SY2012, right? That's the vaccine immunogen.
Except for the one with the typo, the negative Vontrol.
Yes.
So basically...
often used in Transylvanian papers.
These results show multiple antigenic landscapes for broadening G24 neutralizing responses, including highly accessible residues that lie across antigenic sites. That would be the class 1 monoclonals, and 2 residues that are less surface exposed but highly conserved across the G2. That would be class 2. All right, 16 for participant B. They produce most likely in E. coli, but maybe somewhere else. 16 monoclonals. That's 60% of the anti-G24 serum IgG repertoire. And 5 of them that aren't very good, so they don't look at those anymore. The remaining 11 blocked ligand binding to the vaccine immunogen G24, SY2012. 2 of them, VX22 and VX20, blocked ligand binding of 6 additional G2 genotypes. And the neutralization assay confirms that both these monoclonals, VX22 and 20, reduce replication in their cell culture assay. So that's pretty cool. So that's this is indication that, human IgG can neutralize multiple G2 viruses, which is cool.
Yeah, so it looks like they're starting to find a part or an epitope that might bind to these broadly neutralizing antibodies that might be very conserved. And you can think of lots of ways that could be used going forward.
Now, they want to know how these antibodies arose in these people, right? Antibody evolution trajectory analysis. This is the collection of words that Kathy never saw together. IG5ML. So they wanted to find the unmutated common ancestor that served as the precursor for all of these antibodies. And so they say all members of this lineage were found on day 8. They have the same variable light chain, but a differently mutated variable heavy chain, VL versus VH. And they can produce different versions of these in some organism, most likely E. coli. Many words to avoid recombinantly.
Yeah.
So you can swap VH and VL from the ancestor and the different intermediates and find out what the result is. And it's very interesting. When the unmutated VH of UCA1, which was the
universal common ancestor.
The ancestor, thank you, was paired with the conserved mutated VX22VL and UCA2. The antibody not only had improved G212 blockade, but also gained G23 blockade, which shows that the VL somatic hypermutation alone contributes to G23 blockade. I thought that was really cool.
Yeah, light chains matter.
Light chains matter. LCM. You have a T-shirt with LCM on it. Yeah. Well, maybe you don't feel strongly enough. I don't know. So then they also had antibodies with VH sequences corresponding to intermediate nodes in the tree that led to VX22, right? Or diverged clonotypes, and so they can study those, and they conclude that the ancestor of VX22 likely originated from an earlier G212 infection, and then subsequent infections resulted in an altered VL chain or a mutated VL gene that acquired neutralization breadth to G23, and then later affinity maturation of the VH resulted in expansion of the breadth for VX22 and other members. It's pretty cool that they can.
Yeah, and this is important to know, because if you're trying to use this antigen as a vaccine, or if you have a goal of a vaccine strategy of eliciting this antibody, you need to know what types of things need to happen to the B cell in order to make this antibody. Is this a simple somatic hypermutation process, or is it complicated, where only some paths will work, or will lots of paths work? Yeah. And it might be that the reason why this person has this more rare antibody is because their B cells were perhaps infected with a few different versions of this virus that sort of led the B cells down a path to hypermutate in this way. And so if we're going to vaccinate, perhaps the only way to actually get your B cell to go in this route, to go on this trajectory, as they would say, is to give that B cell the same series of epitopes over time. And so maybe you actually have to do 3 epitopes, one after the other, to sort of push the B cell down this weird trajectory and get this amazing antibody that other people don't make. And so thinking about what sort of series of antigens did the B cell see to push it to have these hypermutations is important. Yeah. And there are some sort of very large studies of these types of things that are done in some other neutralizing antibody settings.
More optimistically, it's possible. I think Brianne's explanation is pretty likely that this pathway was how this person got to this point. But it may be possible, if you know enough about the antibodies that you're getting and about the specific epitopes they respond to create an epitope that may not look anything like norovirus, but that elicits this type of response and gets you to the end point without having to go through those intermediate steps. It's possible.
Yeah, but you got to know all the stuff first.
You got to know all the stuff first. This is the groundwork to get to either of those outcomes.
All right, so next they solve the structure of VX22, which they really like, together with a domain of VP1 from the vaccine G2.4 SY 2012. All right. And I want to tell you one thing, just a few things about that. The epitope, so that they can see what the epitope for VX22 is, right? Because you can visualize the antibody bound to the virus, and you know what amino acids it's touching. So interestingly, the epitope is distant from the ligand binding sites, right? We're using ligand blocking assays to measure these antibodies, yet where the antibody binds is far away from it. So they thought, could it be that the antibody breaks up the virus particle? It's so far. Maybe if you broke it up, that would be the same thing. It couldn't bind anymore, right? And in fact, they incubate the antibody with virus particles, and they look at it, and they're all broken. So Petra Levin on TWiV always says, you should always look in a microscope at what you're doing. And you can find out a lot of things. So it's this antibody, and they also see from the structure that the FAB approaches the epitope at an upward angle. And they think as a result, the consonant region projects downward toward the virion interior, which they think either causes substantial steric clashes with a neighboring domain or minor clashes with the heavy chain of the antibody and a neighboring S, and that's leading to breaking up of the virus particle.
Which is cool, because I don't think I've ever thought about an antibody doing that.
Explosion.
Yeah.
And they say the mechanism is disassembly, which we could leverage for designing future immunogens that may have broad protection across G2 genotypes. Okay? So those a little bit complicated, but I will summarize for you. We have a couple of antibodies here. So in participant A, half of the serum repertoire targets two specific epitopes on the virus particle. So these are immunodominant sites, and they probably neutralize by hindering the ligand interaction with the virus particle. And then class 2, VX6 and VX10 broadly blocked all tested G2.4 variants, and they probably neutralized by long-range allosteric mechanisms, not by breaking up the virus particle. Those are from participant A. For participant B, they got these cool monoclonals VX20 and VX22, and VX22, as you saw, neutralizes by breaking the virus particle. So these, they say VX20 and 22 are leading candidates for prophylaxis and therapeutic treatment of norovirus-associated illness. I guess if you had an outbreak in a facility where a lot of older people live, you could give everybody a monoclonal, right, to keep them alive, something like that. So that would be, in addition to a vaccine, maybe you didn't have a chance to get everybody vaccinated, you could just do that.
I also, when I was reading this and got to the end, maybe this is just that I've, it makes me feel old, I guess, because I look at things like Figure 4 and I'm like, oh, and then they just threw in a structure.
Yes.
Like that didn't used to be like a whole paper and a whole thing in and of itself.
While we were on our way to the store, yeah.
Okay, that's the second paper for your. So a lot of activity in norovirus. People are always asking us what's going on with norovirus. There you go.
There you go.
We got a bunch of vaccines and monoclonals. And all I want to say is I hope this continues.
Yeah.
Let's see, it's 4:20. We can do a couple emails. Kathy, can you take the first one?
My screen just went dark. This. Yes. Bronwyn writes, Dear TWiV team, I was very excited to listen to TWiV 1207, Taking a Shot at Dementia, because for once I had already heard about your featured article in an Australian newspaper, which gave some interesting background to the research, highlighting yet again that scientific endeavor rarely runs in a straight line, and you often need a tough hide to be a good scientist. It was great to read that further research has been undertaken with Australian electronic health records, which is awaiting peer review but had similar results. And Bronwyn gives the link for that. I'm hoping the link is not paywalled, but in case it is, I've included the text, which doesn't have the external links below, and provides that. Thank you for making science accessible. Rats, my screen my mouse flew around and my screen went dark, and I may have it pop right back up where I can access where I was if I can find my mouse. Okay, in real time. Thank you for making science accessible. Your message has never been more important. I'm grateful to be living in a country where electronic records can be accessed and science is funded and mostly valued, although we don't have the same resources as the U.S., and cuts to your funding will also affect Australian research. And I'm also glad that through Patreon, I can make a very small contribution, the cost of a cup of coffee, to your work on the other side of the world. And I encourage all your listeners to try to do the same. Thank you also for your moving commemoration of Dixon Des Pommiers' life and work. And again, for all you do. Best wishes, Bronwyn. Thank you, Bronwyn. That's very kind.
Nice.
Thank you.
Alan, can you take the next one?
Sure. Sonrisa writes, hello to all, but Vincent, Rich, and Alan specifically. Thanks for the paper today on the shingles vaccine. Although I am younger than the target population for Shingrix at 44, but maybe I should talk to my doctor about getting it, as I have had the misfortune of experiencing shingles outbreaks twice already in my life. Once when I was only 5, and that was horrific all through my midsection. Yes, they confirmed it as shingles and not chickenpox. I had already had that too. And later at 22, I had a very small outbreak. Anyway, that was a cool little bit of research, and having listened to the recent twin on herpes and Alzheimer's, it was fun putting the bits together. Thanks always for all you and the rest of the Microbe TV team do. I personally love being able to hear interesting science from across such a wide array of topics and connect back between the different podcasts. Cheers, Sonrisa. Thank you very much.
See, young people can get shingles.
Mm-hmm.
Yeah.
I had a student years ago in my class who had, in her 20s, and she had shingles. But sometimes it's because people are taking immunosuppressive therapies for other things, and that can allow early reactivation. And
sometimes bad stuff just happens.
Yeah, sometimes it just happens. So you could talk to your doctor about getting in early. But the problem is that the study was done with Zostavax, and now the shingles vaccine is Shingrix, different. We don't know if it will get the same protection. I don't know.
But I think if you've had a shingles episode, regardless of your age, you probably ought to get Shingrix so you don't get that again.
Okay. Brianne.
Loic writes, Dear TWiV team, I hope this message finds you well. I've been meaning to write you for a while, and the title of your recent episode with the word pluviosity finally gave me the perfect excuse. When I saw it, I thought about the word pluviose and the French history books I read in my teens. I instantly smiled, not only because of the fascinating virology meets rainforest discussion, but also as a listener from Guadeloupe, I was reminded of the French Republican calendar, which replaced the Gregorian calendar during the Revolution, the same folks who gave the world the metric system. Pluviose refers to the rainy month that usually spans late January to mid-February, around my birthday, in fact. Allow me to briefly introduce myself. My name is Loic Dragon. I am originally from Guadeloupe, a French overseas territory in the Caribbean, and I hold a PhD in virology. After a few years as a postdoc in Pennsylvania, then back to Guadeloupe and away from research, I am now working on reconnecting with science, particularly through the world of virology and bacteriophages, which I find absolutely fascinating vis-a-vis their potential for science, health, agriculture, etc. And since there is not much research here in the field, your postdoc is a true inspiration and a key part of this journey back into the topic, virology and other fields like microbiology. For instance, when you mentioned Thiomargarita magnifica in a past episode of TWiV years ago, I nearly jumped out of my chair. That giant bacterium was discovered on the very mangroves of Guadeloupe. I was thrilled that you covered it, and I want to give a special shout out to Professor Olivier Gros, who is my microbiology teacher in Guadeloupe and a key member of the team behind that discovery. It was a proud moment for us here. I also had the chance to meet Dr. Racaniello years ago in Hershey, and I've never forgotten the experience. It meant a lot to me then, and it continues to now, especially as I hear your discussions from my corner of the world. I thought you might enjoy knowing that TWiV has a regular listener from Guadeloupe, proof that your voice carries far and wide, even to places that might not often make the virology map. Thank you for the work you do and for keeping science accessible, engaging, and very much alive. With warm regards, Loic. And I will also thank the folks who, thank the other members of this team who had highlighted the pronunciation. Fortunately, I had seen it right before you did that because I would have been a little lost.
I think we should do a live TWiV from Guadeloupe.
Mm-hmm.
Maybe in like February or
Can
something.
I go? I'm in.
This is lovely letter. Thank you so much. Mm-hmm. And we love to hear from listeners everywhere. So
Yeah.
I would not know that you're listening in Guadeloupe. So it's really good to know this. Yeah, Thayer margarita magnifica, huge bacterium. Huge. The biggest known, I think. Something like that.
Wow.
All right. One more. Charles writes, not sure if this is a question for the regular TWiV or the clinical update. Well, I thought it was a regular. Not a great day in Chapel Hill, North Carolina, 24C, which is nice, but drizzle, which is not. I was doing something a bit dangerous last night, and for the past few days, I've been thinking about the measles outbreak and what would be nice to have to help combat it. I know a functioning HHS, CDC, FDA, NIH would be a good start, but that is out of the question for a few years. Came up with monoclonal antibodies for measles as being useful. The first use would be as a post-exposure prophylaxis. An infected child goes to school exposing a lot of other children. Those exposed, unvaccinated people could be giving a monoclonal to prevent disease. Second, and more complex, give monoclonals for measles to children under 8.5 months of age. See the paper on TWiV 1204. Which is long-term dynamics of measles virus specific antibodies in children vaccinated before 12 months of age. I remember that. That will be traveling to an area with a measles outbreak. I think this would get around the problem of a third MMR shot, and I hope would preserve the favorable dynamics of the measles vaccine given at 12 months. The complication being that a monoclonal antibody could interfere with the vaccine if given too close together. With RFK Jr. and the Trump-Musk administration in charge, we're going to have lots of opportunities to try different treatments for measles and other infectious and preventable diseases. Later, Charles. Yeah, the paper we did on TWiV was all about re-examining the idea of when in the first year you could give MMR, where you can't give it less than 6 months because you're going to have interference with maternal antibodies. But how close to 6 months can you go? And they concluded that 12 months is the right time. It's a long time to wait. It's just part of the problem. I don't know. I mean, a monoclonal could have, in a school situation, but, if you have a school where everyone is unvaccinated, I don't think they're going to take monoclonals because they don't want anything injected into their bodies. Their parents don't, I wish you could fast forward them 15 years and ask them whether they want this vaccine or not, and then come back to the present and give it to them or not based on their preference. Because, a lot of kids, when grown up, would probably
prefer to get this. Yeah.
Yeah. But they're in the hands of their parents. That's the way it works. Anyway, thanks, Charles, for that. Yeah, I don't know what the situation is with measles monoclonals. It's a good question. If anyone knows, I have never seen a paper. So, don't know. I know that did a podcast at Georgia State. I think it was Richard Plemper. Mm-hmm. Where is he, Kathy?
He's at Georgia State.
Georgia State. And he says there's absolutely no market for measles antivirals because the vaccine is so good. Maybe it's the same for monoclonals.
Yeah, maybe.
Okay, let's do some picks of the week. Brianne, what have you got for us?
So I have an article that I saw on CNN that I was sort of excited about. So you may recall from some past discussion of sort of early life on Earth, an experiment called the Urey-Miller experiment, where two scientists, this experiment was in the 1950s, took a lot of the chemicals that were thought to have been found on the early Earth and put them together in an experiment in a vessel and added lightning and showed that they could get nucleotides and amino acids, particularly uracil and glycine. And this was sort of an idea related to how life began on Earth, because some of these compounds are the compounds you need for early life. I've always had a sort of a soft spot for this paper because I think it was the first paper I ever read, because I heard about it in high school and I decided I was going to read it. Not that I understood anything from a 1950s paper, but, I tried. So this article says that researchers actually basically redid this experiment that to show how life on Earth could have started. But they made one fundamental change, which is that they did not add lightning as sort of big exogenous bolts of electricity. Instead, they looked at electron transfer between different types of water droplets, which they call a micro lightning. They call micro lightning in terms of just small electron transfers that are happening between water droplets of different sizes.
I think that's what the rest of us would call static electricity. But
yeah, exactly. And they actually showed that static electricity, micro lightning, electron transfer, whatever you feel like calling it, was actually enough to allow for the production of uracil and glycine from these compounds. And so in conditions that perhaps mimic what was seen in the early Earth, in the early Earth oceans, you can see that some of these organic molecules can be spontaneously produced. And so in a recent paper in Science Advances, they published all of this information with sort of this other, even easier idea. They wouldn't require, the right compounds getting struck by lightning, but is perhaps even more likely something to have happened. And I was just sort of got a kick out of reading about how even these sort of classic experiments are being updated now.
Very cool.
I don't know, the guy in England, Mark Lane, is that his name? He's written a bunch of books on...
Yeah. Nick Lane.
Nick Lane. He thinks this is all nonsense because he thinks life originated down on the ocean floor, And he says, we don't need no stinking lightning or anything.
Yeah, well, see, now you don't. I bet there's static electricity down on the ocean floor.
Maybe.
Sure.
Yeah. Kathy, what do you have for us?
Well, I picked today's astronomy picture of the day. Today is April 11th, and there's always a permanent link if you go down to the bottom. And what it is a cool image that someone caught. They were doing a video, and in just one frame, they caught the International Space Station in the crescent of Venus. So you could see the shape of the ISS. And so as a kind of shout out to Rich, who told us about these ways to see the ISS some years back on TWiV, I put in the link for how you sign up for the NASA alerts for your area. And they have kind of a cutoff. So they're not going to alert you for every time it passes over, only when it's high enough up and for long enough to make it worth your while. And then there's several apps, and the only one that I'm familiar with is called Go ISS Watch. It's at the Apple Store, and there's a user's guide that I put the link in for. And there's similar links for Android and so forth. But I get the alerts from NASA, and then I set an alarm on my clock, and then I open up the app just about the time it's going over, and then it shows me, where to look and where the moon is, if the moon is out that night and stuff like that. So you too can see the ISS. I've probably seen it, I don't know, between 20 and 30 times now. But yeah.
I've never seen it.
I don't know that I've seen it, but now I'm going to
knowingly
find it.
Yeah, I looked, and there's not really any good passes in my area for the next couple weeks, so it may be true for you as well. But set up the NASA alerts, and then they usually come 12 hours ahead of time. And you'll be amazed at how fast it goes. When you first see it, it's like, whoa, that's really going fast. and the app shows you, because it has to do with where it's picking up the reflected sunlight. It's usually a little before sunrise or a little after sunset. But then it doesn't, you're not going to see it necessarily traverse across the whole sky depending on where it is. And so the app will show you it's just about to go dim now, and you're watching it, and then it goes dim and it's gone. Yeah, it's cool. And I always wave to Kate or whoever's she's not on the ISS ready anymore. Yeah.
I've seen it a couple of times. I've also heard it several times. There's a ham radio repeater on the ISS, and hams will contact through. I've tried to make a contact through it, but haven't succeeded yet because I don't have my antennas set up right. And I usually think of it like too late in the process, and then anyway.
In the 50s, when I was a kid, I remember one night we were living in Paterson, New Jersey. My father said, "The Sputnik's going to pass overhead. Let's go see." We all went outside, and there's this light moving across the sky. I think it was an airplane. I don't know, but maybe it was Sputnik.
Airplane.
Maybe it was a Sputnik. Who knows? seemed kind of low to me. I didn't know, but
you needed an app.
Yeah, 1958.
Yes, you
I
need
don't
an
think
app
so.
in 1950s.
Alan, what do you have for us?
So I have an article, a little news article. Dixon would have loved this. So people are probably aware that honeybees, especially commercially maintained honeybees, have been having some problems, dying off for a variety of reasons. And this is a problem because, not so much because of the honey, but because of pollination. They are the mainstay of pollinating a lot of our crops. So this is an article about a different type of bee, mason bees, which are native to North America, and are apparently much better than honeybees at pollination. They don't make honey. They're solitary bees. They don't sting, so that's another nice thing. But you can build a house for these. It's like a house with a bunch of little tubes in it, and they make their nests inside those tubes with some mud, hence mason bees. And you can lure them to your backyard to pollinate your own flowers, and potentially this could be used at crop scales to pollinate fields of crops, and there's an organization that promotes this, and I just thought it was really cool.
I would recommend people read this article because it describes how the bee does its pollination.
Yes.
And it says that it belly flops onto a flower. Sounds great.
And
so the key issue is honeybees are very fastidious and neat. And so they'll get their nectar from a flower and they'll clean off the pollen from themselves and they'll go to another flower. And so they do pollinate, but not really well. Whereas these mason bees are they don't care. They just belly flop on a flower and then go belly flop on another one.
And the article also answered a question I had, which is whether they have queen bees and worker bees. And the answer is no. All the females are queens.
Yep.
So this is really cool.
Yeah.
I have a very short pick. It's a research highlight in Nature, which just gives a little summary of a published paper. It's called Mystery of Medieval Manuscripts Revealed by Ancient DNA. And I have to look at it off to the side here on my laptop because I'm looking at it under the Columbia account. So this is pretty interesting. Medieval manuscripts. This one was done by Elodie Levesque at Panthéon-Sorbonne University in Paris. And they wanted to know what the covers were made of. Because these 12th and 13th century books, from Cistercian monasteries in France, Belgium, and the UK, the books, the covers are often fuzzy, right? And so they took some of this stuff and they did DNA analysis and collagen analysis, and they find that the bindings are made from seal skins. 4 covers were identified as coming from harbor seals. One was a harp seal, and one was a bearded seal. And so this is interesting because most of these animals are from the seas around Scandinavia and Denmark, and the presence of these in places as far away as France attests to an important medieval trade in sealskins. And these pelts could have been purchased from Norse traders or merchants from the Hanseatic League, as they were both active in the region in the time. And when they were first made the book covers, they probably were a pale color, like the modestly undyed vestments of the Cistercians. So I think that's cool. The manuscripts have texts in them, which is important, but what they're made of is also informative.
You can judge these books by their covers.
That's right.
Very cool.
We have a listener pick from Sonrisa who sent us a letter, and part of it was a pick. I hope you take a short minute to watch at least one of this guy's videos. He's a ride operator that pranks his riders. So if you need a laugh, check it out. I didn't look at it because I don't like being pranked on rides. It's scary. I have problems with scary roller coasters. I think we talked about roller coasters last week. I just can't handle the scary ones. I don't know why. All right, that's TWiV 1209. show notes are at microbe.tv/TWiV. You can send us your questions, comments, pics. Tell us about yourself. TWiV at microbe.tv. And if you like these programs, we'd love your support. Microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, bioprof. Barker.bsky.social on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. We have a Microbe TV Blue Sky account if you'd like to go check it out. It's Microbe TV, no dot, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.